Today, we're very excited to have Dr. Ben Jantzen with the president of the Virginia Institute for Invertebrates, and he's going to talk about conserving Virginia's extraordinary invertebrate heritage.
Thank you. Afternoon, everybody. So I am happy to take questions as we go. Just stick a hand up. I don't mind getting interrupted. In fact, I prefer to hear what you have to think or what you're unclear about. But as Ann said, I'm here to talk about invertebrate animals and their conservation in Virginia. I want to start off by saying something about this extraordinary heritage of invertebrates that my title alludes to. And let's start globally, right? So when you think biodiversity, at least among animals, you should think invertebrates. More than 95% of all animal species are animals without a backbone. They are invertebrates. And terrestrial arthropods, so jointed segment creatures, terrestrial, think insects, millipedes, centipedes, isopods. Terrestrial arthropods by themselves outmass wild mammals by a factor of about 30. So not only is there enormous diversity in species, but there's a huge amount of living material in the invertebrates. And they play essential roles in pretty much every ecosystem. The way I framed it here is in terms of ecosystem services of decomposition, nutrient recycling, forming a principal layer between the primary producers, the plants, and things like birds that eat enormous numbers of, say, insects. But of course, that's the sort of vertebrate-centric way of looking at it. If we think about flow of energy and mass, and again, diversity or variety, the invertebrates are the ecosystems. They make up an enormous fraction of what goes on in the living world. And what's true globally is true in Virginia. So my daughters helped me put this collage together some time ago. These are all species, if not endemic to Virginia, then characteristic of it, giving you a sense, just a very small sense, of the enormous variety of animals that fall under this heading of Virginia invertebrates. Things that we all know, like Virginia carpenter bee, charismatic things like butterflies, right? Our state butterfly, the swallowtail, but also arachnids, mayflies, giant water bugs, millipedes, even some extraordinary on the upper left there, flatworms of insane form and just a small taste. If we want to drill down a little bit, because I can't possibly give you a survey in this time of all the major types of invertebrates in the state, I want to look at just a couple that are really special in Virginia. Millipedes is a big one. The Appalachians are a hotspot for millipede diversity, which is why Virginia Tech houses one of the few global areas of expertise in millipede systematics, Paul Merritt's group. And the ones that I've put up here are varieties of just a handful of families of flatback millipedes. These are all cyanide producers. And these are all known from only one or a handful of spots in Virginia. In fact, you know, this one's famous because it was discovered on the Virginia Tech campus. This one, let's see, the upper middle, I like because it's only known from Pandavis Pond. That's it. This one over here doesn't even have a name yet. Jackson Means, who was on the board of the Virginia Institute for Invertebrates, is still describing that species. Yeah.
By any chance was the Hokie one discovered in the old growth forest?
Yeah. I believe that's what it is.
I have a question.
Yeah.
I know they're different colors, but they kind of look alike, and I used to work in entomology, but I don't know what the key character is for distinguishing species and notes. Or do they use name?
So you're right that everybody so this is really mostly Aphiloria, but there's a couple other families represented here. These are all in the Polydesma, flatbacks, and broadly speaking, they've all got kind of the same shape and they're traded more or less the same colors. Most of the taxonomic work is in describing the Dento.
Okay, because I know Palmer, but I've never heard him talk about that.
Yeah, for millipedes, it is a pair of.
That might be DNA.
They use at least 3 lines of evidence when they're doing the description. Geographic distribution, of course, all these are known from what they call spots.
That's partly because it's not going to be for collecting.
That's a huge part, yeah. Exactly. So nobody knows exactly what the extent of the range is, although there is reason to think that it's not merely a sampling issue. That in fact, there are conditions in Appalachia that promote diversification in these millipede clays in particular.
You know, these are mainly older, this genera, they're a lot the same genera.
Yep.
Okay, so they're not, at least they're old.
Yeah, so these are all the same, well, these, yeah, so most of these were described, actually, all of these were described in the last decade. So now that people are taking a sustained look at millipede systematics in the state, in other words, you know, we don't recognize what we don't look at. And now that people are paying attention, we're realizing there's an enormous diversity of species right under our feet that we haven't even named yet. The same actually could be said, I don't have a slide for this one, but Jantzen is also on a group that's doing much the same thing for house spiders. So house spiders, so that, you know, the little gangly-legged fellas that are hanging out in the corners of your house. It turns out one of the most familiar arthropods to all of us, right? You see them all the time. Nobody looked close enough to realize how many species are actually there, even here in Virginia. And they just named a couple species in Tennessee and Virginia and Carolinas. Yeah, so we're a real hot spot in Virginia, Appalachia in general, Virginia in particular, for millipede diversity. Another thing to highlight, I think, is our underground Galapagos. So that's a picture of the Galapagos Islands. This is famous from Charles Darwin's voyages and work, right? Island biogeography in general was a big part of the argument for evolution by natural selection, because islands have some interesting properties with respect to biodiversity. So they tend to be relatively species poor, but relatively rich in endemic species that are only found there, no place else. Island chains, right? They are colonized at a low rate, but colonizers are separated genetically from their original stock and tend to diverge. We have something like that here in Appalachia. It's just underground, not islands in an ocean. What I'm referring to is the karst. We have large cave systems in the state, right? We sit on top of a lot of limestone, dolomite, dissolves, forms, amongst other things, these extended cave systems that partly carry groundwater. And these cave systems act a lot like islands. So if you are a terrestrial cave denizen, you may be effectively isolated from other caves mere kilometers away. If you are an organism that lives in the groundwater, and groundwater organisms actually get around pretty well, you're still isolated by drainage basins, which is what's being shown here. And actually, over here, we see the Powell and Clinch rivers and the Holston Rivers drainage. They're only 20 miles apart. But for the organisms that live in the groundwater of that basin, you're effectively a minimum of 80 miles apart. That's how long they have to travel downstream and then upstream to get back into the other basin. So we have these cavern systems that act like islands, isolating groups of organisms. And there is a rich fauna that is endemic to these caves. Now, this is not showing specifically organisms endemic only to Virginia, although They are largely isolated to the Appalachian forests. These are a variety of freshwater invertebrates, everything from platyhelminthes up there, flatworms, to some snails, to a whole bunch of arthropods, particularly amphipods, which maybe more familiar with in a marine environment, they're kind of a little shrimp, and isopods, aquatic freshwater isopods. Isopods are your woodlice or roly-polies or sow bugs, lots of different common names. We're used to finding terrestrial ones under logs, but these crustaceans also have a variety of both marine and freshwater species. Yeah.
And their title, what does it start go by and time?
Yeah, great question. So,
Sorry, can you repeat that?
Yes.
For the folks online?
absolutely.
Maybe say a little closer to the.
Oh, right, the microphone. I keep forgetting about that. Yeah, Zoom. Stigobiont is you live in the waters of caves, cavern systems. So think of the river Styx. You live in you're aquatic. You're an aquatic cave organism, and the troglobionts are terrestrial cave organisms. Yeah. So just to give you some examples, these are photos provided by Jerry Lewis with the Virginia Museum of Natural History. In 2023, they just finished a big survey of cave fauna in Virginia. And this is just to give you a sense for what these guys look like. Unsurprisingly, most are unpigmented and mostly blind. That's one of those amphipods I was talking about. This is some of the diversity of isopods. So these are aquatic isopods for the most part.
And they breathe in water?
Yes, so these guys in particular, the tricky bit for them is breathing on land, actually. So these are crustaceans, and they have essentially gill plates that they need to keep wet in order to exchange gas. So in water, they're fine. Under a log, they got to stay moist in order to be able to breathe. Yeah, I mean, long and thin. Some of them still retain pigmentation. That wasn't an isopod, sorry, myriapod. And this guy is not a stigobiont, but Jerry sent me this picture and it's adorable, so I put it in there anyway. That little guy is the nosy pillbug. That's an introduced species. You find it all over the place under logs around here. You find it in caves too, which is why he has the picture. This is from the survey, but it's not endemic to caves or even here. It's an introduced species. Okay. Here's something unique to Virginia. This is the Virginia fringed mountain snail. Anybody ever heard of it? It's very small. So these shells are between 2 and 4 millimeters across, all the shells that we found. So far as I know, no one's ever found a living one. We only ever find the shells. And part of the reason is that they are fossorial. Anybody remember what that one means? They live in the ground. They burrow. Don't ask me how a snail burrows. I am very curious myself, but they burrow. In fact, these little 2 to 4 millimeter wide snails burrow up to 6 feet down into the kind of soil in which they live. Now, the kind of soil in which they live occurs in that green patch and nowhere else. They're known only from some bluffs over the New River, and that's it. Nobody's entirely sure if they're only known there because that's the only kind of, it's soil underneath of talus, crumbling rock. Nobody's sure if that's the only place they can live or the only place that we're able to find them when that soil ends up at the bottom of the bluff and we can dig through it and sift for shells. But this is the only range known for the well, unsurprisingly, an endangered, listed as endangered. That's a great question, and yeah, the short answer is I don't think so. I'm sorry, she's wondering if there is a natural preserve specifically to protect the habitat of those snails. They are listed, federally listed as endangered. So they have some protections in principle, but I don't know of any particular preserve set aside for them. Yeah.
Can't hear you, what?
That area does encompass several natural area reserves and certain Clear Lake State Park, but I think there's.
Yes, that's a good thing to point out, is that that big blob there in the bend is Crater Lake, right? So a big part of the state park is there, but the blob is also misleading in the sense that they're only known from the bluffs along the river edge as well.
Yes, they would be hard to say.
Crater Lake itself probably didn't do them any wonders. That's an interesting question too. Yeah. But you know, we don't have to attend only to the exceedingly rare species to appreciate the mind-blowing diversity of forms and lifestyles in Virginia invertebrates. This little fella here, she is a toad-like bolus spider. I admit that until I found one, I did not realize that they lived this far into North America. I assumed this was a tropical species. She has a very strange shape. The color is a little off because sadly this is a preserved specimen that the museum sampled. When she was alive, she looked more tan or gray than yellow. And her abdomen there was, it sort of glistens. And she looks for all the world like a tiny toad sitting on a leaf, hence the common name, toad-like bolus spider. Bless you. The reason they're called bolus spiders, people familiar with bolus spiders? Have you heard this before? You see that sort of tangle of webbing behind her? Female bolus spiders, and only female bolus spiders and only adult female bolus spiders have a funny way of hunting. So she'll sit on top of a leaf all day. At nighttime, she goes to the edge. It's pretty complicated how she makes that thing that's been preserved dangling behind her. It's called her bolus. She essentially makes a wad of sticky thread that ends up on the end of a line that she can pay out. So she sits on the edge of the leaf and pays out a line of thread on the end of which is this ball, right? And she holds it dangling like that with one of her forelegs. She then emits a chemical signal. They are fantastic mimics of moth pheromones. So she can make herself smell like a female moth. They are specific. They can switch their signal depending on the predominant species of moth in the area. So, I mean, that in and of itself is a wonderful mystery. I mean, that's some complex chemistry. She switches the signal. When a male moth comes looking for the female and flutters nearby, she swings that bolus on a line, smacks him with the sticky ball, and then reels him in.
Not see the 8 legs in this picture?
Yeah, 2 of them are hard to see underneath. She has a very large abdomen, and 2 pairs are underneath that large abdomen. Yeah, you're only seeing the front 2 pairs, the long ones that she hunts with. Yep. I mean, I'm sad they pickled her, but I'm the one that found this one. She's about yay big, maybe 3/4 of an inch across, fairly large. Here's this one. I mean, whenever I do this sort of a talk and I want to share with people about diversity of invert form and habit, and you always think about examples to use. And I never have to think very hard. I just go sit on the deck and see what shows up. This guy started pestering me when I was working outside a day or 2 ago. It is a peacock fly. These are not uncommon. They are not highly localized. They occur all across North and South America. They've been introduced accidentally recently to Europe. But they are very odd little flies. they get their name from the way they're holding those wings, right? So it's got its wings, you know, the little body here, its wings are held up perpendicular to the body. And not only do they hold the wings up when they land and do their display, but they do kind of a little dance. So they wiggle the wings around and they shuttle back and forth. And the wings are really striking. It actually caught a little bit. I used this photo because you can see the blue reflection in the upper right. They shimmer blue as it wiggles those highly patterned wings. This is a type of picture wing fly. So there are others in the family that have similar patterning, but none that reflect like this. It's got this beautiful blue reflectance. Now, why is it doing that? If you look up the peacock fly, I encourage you to do so, you'll very quickly, if you find anything at all, you'll find a couple of articles talking about it being like a peacock, right? So the male peacock has this elaborate display for sexual selection, right? He's trying to impress the females. It's a display the male puts on for a mate. But in peacock flies, both the males and the females do the little dance. So if you look a little bit harder, you'll find some articles suggesting that this might in fact be an instance of symmetric mate selection. So both males and females are being choosy in the species. But if you press a little harder, you'll quickly realize that nobody actually knows the answer for sure, because nobody's seen them mate. We see them do the dances, but we're not entirely sure that the dance really has to do with mating, because there's no study on them actually selecting a mate in the process. So it's a guess, but it's a fascinating behavior and it's beautiful to watch. Oh, that's a good question. I'm not sure what the adults eat, actually.
But they're trying to.
Oh, no, they're not biting flies. Sorry. Their size compared to a housefly, they're about the size of a typical housefly. Maybe a little smaller. They're more gracile, they're thinner. So they normally do this dance on dead logs. It decided my aluminum furniture looked like a dead log. Apparently. It was really adamant about it too. It, you know, when I was trying to take pictures, it would fly off and I frightened it, but it would just come right back and start dancing again. Yeah, I think they're beautiful. How about this? Anybody seen one of these in their yard? You seen one? This guy's pretty unusual. If you found this guy, there's nobody else that looks like it around here. This is the so-called monkey slug, Phobetron pithecium. Anybody have a guess what it really is? It's not a slug, and it's obviously not a monkey. It is a caterpillar. Yeah, it is a caterpillar of what's also known as the hag moth. Yeah, why does it look like that? What does that look like to you? Is that evocative of anything? Glory, yes. Yeah. A spider. So the going theory is that what these caterpillars are mimicking, other slug caterpillars, other caterpillars in this family do not look like this. They look weird, but they don't look at all like this. The going theory is that this is a mimic of specifically tarantula exuvia, so the shed skin of a tarantula. Now you might be thinking, that's odd, I don't have any tarantulas, and you're right, but Phobetron, that genus, is mostly tropical. So most of the other members of the genus live in the neotropics, where there are plenty of tarantulas. And we also have to remember that most of the birds that would eat them here overwinter in the neotropics, right? They're spending a good chunk of the year in the environment where there are tarantulas that can shed exuvia. What's even more crazy, though, is that the adults are also mimics. If you've seen a hag moth, what did it look like to you? So that I should have brought a picture of the adults. The adults are pretty wild looking too. The females look like bees with full pollen baskets. They have tufts on their legs that look like blobs of pollen. So the females are pretty clearly bee mimics, but the male adults look like something else. The male adults are pretty clearly wasp mimics. So you have one species that mimics three things depending on its age and sex. And this one is in a dogwood tree in Blacksburg here. So they are here in Virginia. Here's another one from just a couple of days ago that I thought I'd put in there because it's also fascinating. So this is a very striking wasp that you may encounter at this time of the year. They usually show up in late summer. The adults do. This one is a female. The males of this wasp species do not have those long prehensile abdomens. The reason the females have those wild, if you watch them, it's crazy. I mean, it looks like they move them around like an elephant trunk. It's ungainly when they fly, but when they stand and manipulate that abdomen, it's really quite dexterous. They use that abdomen to parasitize, or they're parasitoids of June bugs, June beetles. So they use that abdomen to probe into the soil and find a grub and lay an egg on or in the grub. The males have a much shorter abdomen. Now, this is the only species in the family here in North America. It used to be a much more diverse group, I mean, in geological past. And this species is another mystery. It lives throughout the Americas, so from Canada to Argentina, with the exception of a little strip through the middle of Mexico. The populations of the South are more or less equally mixed, male and female. You find male pelicinids, you find females. In the North, males are seldom, if ever, found. It's almost entirely female. Why is that? How is that? The why is hard. The how, there's a lot of speculation. Maybe they are parthenogenic. So that's when you can lay an unfertilized egg that then develops into an adult. So, lots and lots, aphids are parthenogenic. They go through different reproductive stages where they'll either reproduce by cloning themselves in parthenogenesis, they'll just lay eggs that grow into females, or they'll produce males and engage in sex at the end, usually at the end of the season or when they've used up a resource. So maybe these wasps can do that. Nobody really knows if that's the case. There's some strong evidence for it. Actually, the only thing I was able to find past the 1998 paper that pointed out the weird distribution of sex ratio in these wasps was a student's thesis, an honors thesis, as I recall, at Canadian University, where they looked at some of the genetics. And it looks like there's enormous uniformity in the northern population, which is what you would expect if they are in fact reproducing by parthenogenesis. So it may be that the northern pelicinid wasps just gave up on sex. Okay. Well, that was meant to just give you a taste of the kind of invertebrate diversity we have here in Virginia. I want to talk a little bit about conservation and why we should be worried about it. And that means, unfortunately, talking about decline and threats. This has become a very famous figure from a scientific paper. This is from the German Krefeld study in 2017 that kicked off headlines in the New York Times talking about an insect apocalypse. What it's showing is the mass of flying insects captured over fields in a series of, I think, 27 locations across mostly northern Germany. Actually, the plot you're looking at here runs from 1990 to about 2015. The study itself spanned actually closer to a century, the full study. And what you see from 1990 on is a bit of a disturbing downward trend. It's a lot more disturbing when I tell you that this is plotted in a fancy way. That vertical scale is logarithmic. Yeah, meaning that, you know, we start somewhere around a mean of 7 on that plot, and you end somewhere around 1. Yeah. It amounts to a 75% loss over that span of time. And that's not a particular species. It's not a particular collection of species. It's not like they were just looking at butterflies or flower flies. This is the total biomass of insects on the wing over these regions. Sadly, something like that trend is showing up robustly across the globe. It's not the same everywhere. It's not the same for each group. It's very hard to say things that are universally true of any group of organisms that diverse. Some varieties of insect in particular are increasing in number, but collectively, if we look at these aggregate measures, mass, abundance, we see this kind of steady loss. Why do we see this kind of steady loss? It's a long list. It's not any one thing that we can point to, but big ones are habitat loss. When you convert most of the land area of a continent to agricultural production, urban areas, suburban areas, you lose large tracts of habitat. Unsurprisingly, you lose numbers of the organisms that depend on it. Similarly, the habitat that's left is a bit deceptive. It's not the total area that matters when the habitat is fragmented. That is to say, when suitable patches of habitat are broken up by regions of unsuitable habitat. So while, for example, the forest cover of Virginia may remain an impressive fraction of land area, certainly not what it was before we started carving it up, but impressive nonetheless, that's misleading on account of how divided that forest really is. And I'll show you something pertinent to that in just a moment. Another big one on the list, so I'm trying to get the biggest drivers here, are invasive species. That is to say, human-induced mixing or introduction of varieties of organisms into a range or an ecosystem in which they have not historically occurred. This can have a mixture of effects, but it's a huge one for a couple of different reasons for invertebrates, and I'll say a little bit more about that too. I won't say much about it in detail, but pesticide use is an enormous one. And when I say pesticides, I don't just mean things like insecticides designed to kill particular sorts of invertebrates. I mean herbicides and fungicides as well. Herbicides and fungicides in combination are extraordinarily lethal to insects. Light pollution. We don't always think about this one. It's on my list of big drivers, and that's not an accident. It's not an incidental thing. More than half of insect species are nocturnal. Light pollution is a huge deal in terms of disrupting reproduction, well, all daily activities of a nocturnal organism, but particularly organisms that take sensitive cues from things like moonlight and stars, or that have been fine-tuned. I mean, a very good example would be the mayflies, right? So mayflies historically occur in enormous numbers. Mayfly emergences overwhelm predators with huge quantities of emerging adults trying to lay eggs for the next generation. Mayflies have been around as an order, more or less in their present form, for more than 250 million years, and they are exquisitely adapted to do what they do. And one of the things they have to do, for example, in their short adult phase, is find a place to lay their eggs. And they want to lay their eggs in water. How do you do that in the dark? Well, surfaces of water are polarized in their reflectance. That's why we wear polarized sunglasses, right? Takes the glare off of passing cars. Light bouncing off of a flat surface, like a stream or a pond you want to lay your eggs on, is polarized. And that's true whether it's moonlight or sunlight. So to a mayfly, a creek running through a dark forest looks like a bright ribbon in a dark background, and it will head for that to deposit the eggs. Unfortunately, wet roads, automobiles in parking lots, windows on buildings all look the same way. And enormous numbers of mayfly eggs are expended at present under streetlights on pavement that obviously can't serve as the next generation. So light pollution is a huge one. Yeah.
From the perspective of birds, what's your opinion on balancing invasive species at bay and using, herbicides, like just cut it and eat the stuff like that?
Yeah. So, of course, it's not going to be a simple answer, right? There are, given that all of these things on my list interact with one another, and in fact, we'll just throw climate change on there as well. It's not like, you can give a simple answer for all cases. Oh, just don't use pesticides at all. Precisely for the reason you said, right? In fact, I don't know why invasive species. Yeah, I'm going to show some plants in a second. It does matter how you do it. So localized applications, like you just said, cut and paint, are different from broad area, broadcasting of pesticide, particularly in agricultural settings. So insofar as we need to do it to mitigate another ecosystem disruptor, it's just important to do it in a way that's conscious of the knock-on effects. You don't want to make it an even worse driver than the thing that you're addressing. And a lot of times with respect to inverts in general, it's about localizing the toxin that we're talking about. Yeah. In particular, even when we're painting, we got to worry about, how broad of an area of soil is ultimately contaminated. How close are we to waterways such that, this is getting into the groundwater or into streams. And then it's just a balancing act. Yeah, that's a great question. Okay. Let me just say a little bit more about some specific kinds of I'm going to look at habitat fragmentation and invasive species. So this is some work I'm doing with a couple other folks at Virginia Tech, Dr. Shao and Will Jones. We are going back, and 20 years ago, there was a great paper called How Far to the Nearest Road, where somebody a team used some early GIS systems to answer that question for the contiguous United States. If you threw a dart at the US, how far would you be standing from the road? We've gone back and we're looking at how that answer has shifted over the past 20 years, as well as some other aspects of it. And we have some preliminary results just for the state of Virginia. So what you're looking at there for the whole state, those colors are actually colored dots. The size of the dot is indicating distance, as is and the color is indicating the ecosystem type. So this is zooming in on Montgomery County. So that red shape in the middle, that's Blacksburg at the top and Christiansburg at the bottom. Red meaning you're in urban area, hardscapes mostly. And those are little bitty dots. You don't get bigger distances to roads till, unsurprisingly, you wind up in the national forest, for example. But don't worry about the map. I'll tell you what the upshot is. Half the time, or at least half the time, half the time or more, you're within 121 meters of a road. So if you threw a dart at Virginia and asked, how far am I from a road? You should expect the answer to be 120 meters or less. Now, maybe I don't know, that sounds really close to me, but it's worse if you think about the average edge effects when we talk about habitat fragmentation. Having a little clump of forest is not like having a big forest, right? So if you look at a little clump inside of a big forest, its ecological properties are distinct from a little forest inside, say, a meadow. You have what are called edge effects. So you have different organisms, you know, because you have different light, different humidity, different chemistry on the edge of a forest versus the interior. Edge effects are, it depends on the organism you're talking about. It depends on the kind of effect, but, you know, 10 to 100 meters is the typical range that we're worried about. But if the nearest road is always 121 meters away, that means most of this habitat is edge. It's all edge. Okay, what about invasive species? So, oftentimes we think of the invertebrates themselves as the invaders. And that's true. We've moved an awful lot of invertebrates around the world, but invertebrate communities are just as subject to the impact of invasive species as anything else. So here's one particular story I decided to tell since I found this guy yesterday. That's a Cecropia moth, caterpillar, obviously. Cecropias are the biggest moths native to North America. You know, they've got the deep maroon fur, maroon, white, and black on the abdomen, this deep maroon on the thorax, and those beautiful brown, white, and sort of gold wings. They get about a 6-inch wingspan. Yeah, they're big, they're hefty. And so, of course, their caterpillars are titanic because in that family, the Saturniidae, the adults don't eat. The giant silk moths, the adults don't eat. So the caterpillar has to pack away enough resources to become the butterfly, produce eggs, fly around and find a mate, and then fly around and lay the eggs, all without taking on additional energy. So their caterpillars are truly massive. This one's about 6 inches long and like a sausage. Yeah, huge. Cecropia abundances have been plummeting. Partly because of all the reasons, well, no, we suppose partly because of the drivers that I already listed, but there's another more imminent threat for Cecropias, and it's an invasive species. And it's one that we brought here on purpose. We brought it here on purpose to deal with another one that we had brought here on purpose. So this starts with Lamantria dispar, the spongy moth, which used to be called the gypsy moth. That was introduced accidentally, well, sort of accidentally. It was brought here on purpose to try to breed a better silk moth, and it was released accidentally in the early 20th century. You know, it ate our forests. And in order to control Lamantria, we intentionally imported a tachinid parasitoid, so a kind of a wasp, not wasp, a fly, a dipteran, a little tachinid fly. And that tachinid fly is a parasitoid, but it's a parasitoid of a broad range of lepidopteran hosts, and it really likes Cecropia. And it seems that is, you know, mortality due to this tachinid wasp seems to be the principal cause of the most recent precipitous decline in Cecropia numbers, which is really sad. They're beautiful moths. I am actually not completely sure because I found this yesterday as the sun was going down, and I didn't get to go back yet and look at the tree. They're generalists. They prefer maples, but they'll eat a wide range of trees. So I'm very curious myself, but it got dark. That could be, yeah. Yeah. See, this looks bright, but this was done with a flash. I couldn't see the tree. I couldn't see much of the tree in the dark. Yeah. It does look like a pawpaw, doesn't it? There was actually about 20 feet away from this one, on the same type of tree, not the same tree, there was an imperial moth caterpillar. So nearly as big. I don't know what it is about that particular stretch, but it's home to some huge silk moth larvae. Okay. Oh yeah. I told you Appalachia is a hotspot for millipede diversity. This is true, but this is a serious threat to that diversity. So those are the invasive Asian jumping worm. You may have seen some of these. I found one a couple days ago. Well, a bunch of them. It's kind of hard to miss because they shoot out of the ground. And I mean, I put up the caption here. This is from US Fish and Wildlife Services. I put the caption up because it had a bunch of their common names: Alabama jumpers, Jersey wrigglers, wood eels, snake worms, and crazy snake worms. And if you watch them, I mean, they're all apt. They do look pretty crazy. I mean, they pop out and they flop around, and it's very intimidating. Unfortunately, they are voracious and extremely fecund, and they are denuding, of all things, the principal layer of decaying leaf matter on the forest floor that all those diverse millipedes have evolved to process. So they are, of course, it's not just millipedes. They're wiping out a layer that is home to thousands of, per square meter, you're talking about thousands of things like deplorans and springtails. So these are other arthropods, but also millions of nematodes. So roundworms that are make up these rich communities of decomposers, hundreds of species. So these organisms are all in competition now directly with this Asian jumping worm. And it's really astonishing at how clean they're able to just scrape a surface. I mean, unfortunately, they're getting more and more common. Yes.
Do they move off? Or is it like, no?
Yeah, that's a great question. So I don't have any particular study to point to, but I can say that, you retain a sort, kind of like a seed bank. You retain a bank of nematodes to sort of reestablish a community like that, and likewise for fungi and so on. But it takes a while, especially on like the thin soils of our oak forests here, to build up that layer in the first place. What matters is that you've got the humidity that the decomposers need to survive. So most of those arthropods I mentioned, they can't live on a dry surface. So, part of me is hopeful that if leaf litter is allowed to build back up and somehow the worms are kept away, that you can reestablish that community relatively quickly. But it really does require some study, because it's also possible that you can't get past the dry phase to bring these decomposers back. Yeah. So big question mark, I guess. I'd like to know. Oh, and I did promise to mention plants. So, what's the biggest determiner of insect diversity? Plant diversity in an area, yeah? On the left there is a picture of a remnant grassland on Grassy Hill. And it's sort of accidentally preserved under the power line right away. And you see the little DCR sign there saying, do not spray. There's some collaborative efforts going on with the power company, but there are hundreds of species of plant in the picture on the left. On the right is a river of stiltgrass. That's on top of Brush Mountain, goes for 4 or 5 miles. And that picture is even deceptive. It's just below my waist for miles and miles and miles. And it's almost entirely stiltgrass. None of our lepidopterans live on stiltgrass. So this is a desert on the right. This is a rich, diverse habitat on the left. Okay, so what can we do? Well, I made you guys sort of a brief list. We could spend a session on any one of these. I kind of reduced these to slogans, but I'm hoping that, if you're interested, you can ask me. I can point you to other groups, etc. But let me throw some out there. One thing we can do if we're thinking small, like our own yards, leave the leaves. Leaf litter is essential components of invert communities. It is the primary habitat for a wide range of decomposers. It is an essential reproductive stage for things like fireflies. absence of leaf litter is a big problem for firefly abundance. There are all kinds of organisms that need the shelter and the humidity preservation of leaf litter to survive. So if you want to increase the diversity of invertebrates on your property, leave the leaves. Don't rake them up and send them away. Plant natives. I know there's a bunch of master naturalists and master gardeners in here. You know, I wanted to make sure I got this one on there. I want to stress that if we're thinking about invert diversity, some species, native species, have a far bigger impact than others. And in particular, you know, I guess I'd also say it's not just about pollinators. Like, don't get me wrong, I love pollinator gardens and the diversity of adult foragers it brings, but we also have to think about host plants for larval stages. You have to think about the many orders of insect that aren't lepidopterans or dipterans and aren't primarily taking their food from flowers. And if you think in this broader way, see it, pretty Native Plant Society, shout out there to you guys. You know, what kinds of plants am I talking about? For instance, oak trees, oaks over some other tree choices because of the insane range of, for oaks, Lepidoptera, for example, that use that as a host tree. Hundreds versus a handful. Here's one that people don't often think about for your yard. Vary the terrain a little bit. You really don't have to do much. You know, we all tend to have graded yards, right? Where somebody literally took a grader, scraped it flat, and then we planted grass on it. If you vary the terrain, if you will allow there to be depressions and mounds, if you allow there to be relatively bare patches, shaded patches, this makes an enormous difference in the number of microhabitats that your yard offers to invertebrates. It's astonishing how much, you know, a depression, the extra humidity of a depression can make a difference in what's able to live even in a grassy front yard. So vary the terrain. If you want to add a water feature, so much the better, right? Having water available, particularly if you have significant variety of plant life around the water source to provide shelter for the organisms that would otherwise be using it. But even something as small as, you know, really digging a few holes, making some depressions and mounds to break up the terrain can make a big difference. Here I'm thinking on a little bit of a larger scale. Yes, both are yards, but I'm thinking of, for example, town managed properties like Heritage, where we have these big hay fields. If we can reduce mowing frequency and time the cuts better with the life cycles of the inverts that are using those fields, we can make a difference. So, you know, I'm always sad when they go and they mow the field down just as all the monarch chrysalises are dangling from the grass, right? If we just waited another month, you know, most everything is done with that phase and is either hibernating lower down under the mass of dead grass or has flown off, etc. Yeah. Well, I'd say October for here. I've been trying to get a little bit of a pilot study done. This has actually been done in a number of places in Europe at comparable latitudes. And so there are some more precise dates you can give there. Nobody's really done it here. So if I'd have to guess, I'd say October. Certainly, you know, a lot of these cuts are done late August, early September, exactly when those chrysalises are sitting there. Even just a couple more weeks would give a longer amount of time for those lepidopterans in this case to complete a life cycle. Lights, right? I talked about light pollution. I put turn off the lights, that's a slogan. There's more than just that we can and should do, right? So changing the spectrum and intensity of street lights, just putting reflectors on street lamps to make sure that light is going where we actually want it and not elsewhere, not generally up into the sky, right? Not at other angles. Attending to the intensity of lights, especially along forest margins. You know, I I know it's the fashionable thing to do to have these strands of yard lights out, you know, I don't even know what they're called, like these garden lights. They have these big round light bulbs. I'm seeing them I've started seeing people set them up in campgrounds, which makes my head explode. Like maybe we don't need to do that all the time at any rate. There's no reason to leave that on all night long. So when possible, turning off lights, and when not possible, ensuring that we are using the light for the job that we need and not spilling any extra we don't need generally into the environment. And, you know, don't spray, that's the other slogan. So broadcasting pesticide is almost never the right answer. Paying someone to come and poison all the wasps in your yard, you know, we can argue about whether or not that's a reasonable thing to do anyway, but insofar as it is, killing everything else along with them is not great. So just, you know, as far as possible, restrict the general broadcasting of pesticides. And I don't just mean insecticides, I mean generally fungicides and herbicides as well. Nope. That's the full list. I wanted to put this one on here at the end because I want to encourage people to think about what's actually in their neighborhood. I'll keep forgetting Zoom. VII has started working with a number of groups in the state that focus on preserving our remnant grasslands. So Virginia does have remnant grasslands. They have an interesting and complicated history, but these are ultra diverse patches of vegetation. A couple of the figures that get thrown around in one study plot, 100 square meters, 120 species. In another of the preserves, around 50 acres holds something like, oh no, I'm sorry, it's around 90 acres, holds 550 species. These grasslands are extremely diverse. And almost certainly, this is part of why we're getting involved, they hold equally diverse invertebrate communities. Problem is that, you know, they're vanishing. They're getting plowed over, paved over, and part of the problem is that people just don't even recognize what they have. So just when I started working with some folks at the Clifton Institute, for example, that week they had lost this wonderful multi-acre patch that was next to a high school. So it was not only a great preserve, it was a great learning resource, but they drove construction equipment right over the patch to build an extension without warning anyone, and the patch is gone. All I'm saying here, just briefly, you can reach out to some folks at like the Piedmont Discovery Institute or the Clifton Institute. And if you've got, you know, a patch of grass, maybe you're near a power line cut, this is a likely place to find such a patch because those cuts have been utility right of ways for more than a century in some cases. If you know of a particularly rich field, it's worth reaching out and asking them to take a look and tell you, hey, is this maybe a remnant? The number one thing for preserving remnants is avoiding plowing. You can bring back a remnant in the face of all kinds of perturbations, even driving construction equipment over it, just sort of crushing everything, as long as the soil hasn't been turned. Once you turn the soil, it's exceedingly difficult, if not impossible, to bring back that ultra diverse community, regardless of how you seed it. That's why I wanted to put that one on there. Okay, so in the last minute, I know we're a little bit over. I just wanted to say something about the institute I represent. So we have a 3-part mission of monitoring, witnessing, and conserving the invertebrate life in Virginia. When I say monitoring, I mean doing the long, difficult work of figuring out how populations of invertebrates in the state are actually changing. Because insects, for example, have such variable annual populations, the kind of work observation that it takes to know if there is a long-term trend you should worry about are observations over the span of decades. And so we're building observation sites across the state to get the data that's just not there and that we're all realizing we really wish we had 20 years ago. when I say witnessing, we are supporting fieldwork in natural history. So all those questions that we had about, why is the peacock fly doing that in both males and females? Why do they both do the display? How is it that the pelicinid wasps are all females in the north, but mixed in the south? Those kinds of questions are numerous for any invert species you care to pick out. Even the most common and familiar of species, generally we know next to nothing about it, the way it makes its living, and the way it reproduces. So we're funding work to do that. And lastly, of course, we're working on conservation efforts through combinations of donations and easements, stitching together corridors between prime habitat patches for invertebrates. And for inverts, unlike for vertebrates, those corridors can be 10 meters across instead of 10 miles across. And, I'll toss a list up there. There are ways to help us. Of course, we're always in need of financial support, but we'd be really interested to hear from you if you'd like to volunteer time, or if you have property that you would be interested in contributing a conservation easement on, or even property you'd be interested in donating to our monitoring program while we're still setting up our stations across the state. I want to thank some folks who helped me out here. So Will Jones, in particular, for those road visualizations, my board of directors and my daughters for the graphics. And thanks a lot to Jackson Means and to Jerry Lewis for providing those great photos that Jerry took of those stigobionts and troglobionts. And thanks to Sustainable Blacksburg for hosting me. There's some contact information for me. I'm going to leave a little stack of cards over here. If people would like to take one, please do. And I look forward to hearing from you, and thanks for your attention.
Well, in the name of thanking, we want to thank you for
Oh.
coming and present you with a sustainable
Oh.
black bag.
Wonderful, thank you.
Absolutely.
Very cool.
Outside.
Yeah, that's awesome.
We're in the 2020 conference.
That's awesome, thank you.
That didn't happen.
Yeah.
Can you go ahead and repeat the question?
Yep.
I don't understand why your institute is part of the invasive species group.
So let me clarify. The question is why the Virginia Institute for Invertebrates is part of the invasive species group. The short answer is we're not. I am. And I was kind of wearing two hats being here. I didn't mention that at the beginning. I am president of the Virginia Institute for Invertebrates, but I'm also a member of Virginia Tech's Invasive Species Collaborative. There's overlap in goals and aims. So like I mentioned, invasive species are very much a problem for invertebrate conservation as much as conservation of any other sort of biodiversity. And so, I do work with the collaborative with that focus in mind.
Okay. Where is it on campus?
So the Virginia Institute for Invertebrates is not associated with Virginia Tech. We are an independent nonprofit. We have a 6-member board and they're spread throughout the state. We have ties to the Virginia Museum of Natural History, or at least a couple of our board members are there. We have folks out at Christopher Newport University, some board members here at Virginia Tech.
Sounds like you're associated with Tech.
Yes, yeah, no, I'm glad to clarify that. We are independent of Virginia Tech. We're an independent nonprofit. But I personally am in, I'm at Virginia Tech and part of the collaborative. And then I also,
We have one question online.
Yeah.
Solar panels installed so as not to destroy habitats.
yeah, so that's tricky. Yes, sort of. You can exchange. So, if we're replacing, usually you're looking to do a solar panel installation in Virginia. We're talking about areas that are already deforested. So oftentimes converted agricultural land, for example. In which case, with some planning and effort, which amounts to an additional cost, it is possible to maintain at the very least the margins of solar farms as relatively rich grassland communities. And there are a number of projects trying to do that under the panels themselves. So, you know, if the solar project involves paving, you know, if we're talking about, I assume we're talking about on the ground, because on the roof this wouldn't be relevant, but if we're talking about projects big and small on the ground, if we're laying down gravel and essentially making a desert, then that's just straight habitat loss. But there are a number of projects that allow plant growth underneath the panels in the farm as well as on the margins. And insofar as that is maintained, that can be a relatively rich grassland community. And that's actually working with some of the major power projects here to use their margins for that purpose is something that we're trying to do in conjunction with some of the folks I listed interested in remnant grassland preservation.
That was really fascinating. I think if we have any more questions, maybe if you're here.
Sure.
Come up to ask them. But thank you again. That
Sounds great. Thank you.
Lots of thanks from the online folks.
Thank you all.
Okay.